So the next topic that we're going to cover in this class is cellular respiration and fermentation. So previously in the course, especially during energy enzymes and metabolism, we talked about the importance of ATP for a cell. So we described it as being this energy currency that the cell can use for various things. So the cell makes ATP and it then can use that ATP to pay for various things that require energy inside the cell. So it's a form of energy currency within the cell. And what we're going to talk about today is specifically how do cells produce that ATP. So we're going to see that both cellular respiration and fermentation are pathways that produce the ATP that the cell can use for those different energy requiring processes. So the first thing that we're going to do before we get into the details of each of these pathways is we're going to compare and contrast the following things. So there's actually two forms of cellular respiration. One form is called aerobic cellular respiration, and then the other form is called anaerobic respiration. So those are both types of cellular respiration. And then we'll also talk about fermentation. And we'll talk, there's actually many fermentation pathways, but we're going to focus on two. One that occurs in our bodies, and then one that occurs among other organisms that occurs in yeast. But we're just going to start by comparing and contrasting these things in terms of what they do and what they produce and how they do it. So again, the one thing that all three pathways have in common, so aerobic respiration, anaerobic respiration, and fermentation is their goal. So they all produce ATP. So aerobic respiration. Anaerobic respiration. Produces ATP. And finally, from the same goal, it produces ATP. Okay, so we said that producing ATP is endergonic, so meaning that it requires the input of energy. And the source of energy for all these pathways is organic molecules. So in each of these pathways, you're going to break down an organic molecule, and during that breakdown, that's going to release energy that the cell uses to make ATP. So the energy that's required for these processes comes from breaking down organic molecules, and they all do that. So in your bodies, some of the food that you eat is broken down in aerobic cellular respiration and used to make ATP for your bodies. And also in your muscle cells, you have a certain type of fermentation pathway that can take place if you're exceeding the oxygen requirement for aerobic respiration, and you can also use food molecules in that process. Okay, so those, both of those things, all three pathways have in common. One thing that makes them different is whether they require oxygen or not. So aerobic respiration, which you could probably guess from its name, requires oxygen. And so aerobic respiration cannot happen in the absence of oxygen. Okay, so if there's no oxygen, then aerobic respiration, that pathway shuts down. Anaerobic respiration does not require oxygen, which means it can occur in the absence of oxygen. And fermentation also doesn't require oxygen. And so it is another process that can occur in the absence of oxygen. Okay, and then the last comparison that I'm going to make is whether an electron transport chain is going to be used in the process. So if you remember back to photosynthesis, we talked about electron transport chains. And in general, what they are is they're a series of components, and they pass electrons in a particular order. So component 1 passes the electron to component 2, 2 to 3, 3 to 4, and then depending on how many components make up your electron transport chain, they're just going to keep on passing electrons. And you're going to use an electron transport chain, which I'm just going to abbreviate as ETC, in aerobic respiration. You're also going to use an electron transport chain in anaerobic respiration, and then in fermentation there is no electron transport chain used. Okay, so aerobic and anaerobic both are going to use a electron transport chain, and fermentation does not use electron transport chain. Okay, do you guys have any questions about the differences or similarities between the three pathways? Okay. So we're going to actually spend most of our time in this topic talking about aerobic respiration. That's going to be our main focus. And then we'll briefly mention some things about anaerobic respiration, and then spend the rest of the time talking about two specific fermentation pathways. So in eukaryotic cells, most of aerobic respiration, oh, I took the picture out. So I'll just mention it. In eukaryotic cells, most of aerobic respiration is going to happen in the mitochondria. Okay? So just to review, in terms of the mitochondria, they have two membranes. They have an outer membrane and they have an inner membrane. And there's also two regions that you should be aware of in a mitochondria. One is called the intermembrane space, and the other is called the mitochondrial matrix. So if you remember, the intermembrane space is the region between the inner and the outer membrane. So the space between the outer and the inner membrane is the intermembrane space. And then the space that the inner membrane encloses, that's the mitochondrial matrix. Okay? So it's just important to be aware of, well, it's really important to be aware of the inner membrane and then also those two regions. We won't be mentioning the outer membrane at all, but we will be mentioning those two regions along with the inner membrane. Okay. Also, when we discuss these different topics, we're going to talk about, we said the goal is to make ATP. And there's actually two methods for making ATP, so these are two different processes for producing ATP. One is called substrate level phosphorylation, and the other is chemiosmosis. Okay? And in aerobic respiration, both take place, but most of your ATP gets produced through chemiosmosis. Okay? So in terms of what happens, in substrate level phosphorylation, this is rather simple. All you do is you have an enzyme, and the enzyme takes a phosphate group off of an organic molecule and attaches it to ADP to make ATP. Okay? So in substrate level phosphorylation, an enzyme is going to take a phosphate group off of one molecule and then directly add it to ADP. And then in chemiosmosis, what we're going to see, this is a little bit more complicated. In chemiosmosis, this is a process for making ATP in which you use a electrochemical gradient as the energy source. Okay, so if an electrochemical gradient is used as an energy source to produce ATP, then it's called chemiosmosis. Okay, and when we discuss aerobic respiration, we're going to say that on average you produce 36 ATP. Okay, so in aerobic respiration, on average you produce 36 ATP. And you only produce 2 of those by substrate level phosphorylation. Okay, so that means the other 34 are being produced through chemiosmosis. Okay. When we discuss aerobic respiration, we're going to break it down into 4 stages. The first stage is called glycolysis. The second stage is called pyruvate oxidation. The third stage is called the Krebs cycle. Alternatively, you can call it the citric acid cycle, or you may have heard of it called this. And then the fourth stage is oxidative phosphorylation. And I made a mistake when not really that important, but I told you that 2 ATP get made by substrate level phosphorylation. It's actually 4. So 4 get made by substrate level phosphorylation. The other 30 get made by chemoosmosis. So 4 for substrate level phosphorylation, 30 for oxidative phosphorylation. 32. Can't add. 32. Okay, so first thing I'm going to do is talk about where each of these things take place. So glycolysis happens in the cytosol. Pyruvate oxidation happens in the matrix, and that's specifically the mitochondrial matrix. And then the Krebs cycle also happens in the matrix. And then oxidative phosphorylation occurs at the inner membrane of the mitochondria. Okay, so with the exception of glycolysis, the last 3 stages of aerobic respiration all occur in the mitochondria. So again, pyruvate oxidation and the Krebs cycle are going to happen in the matrix of the mitochondria, and then oxidative phosphorylation happens at the inner membrane. Okay, so also important to know is how many ATP each of the processes produce. And when we talk about how much ATP is made, we're going to focus as glucose being the energy source. So we're going to focus on glucose being the molecule that gets broken down to provide energy for the ATP production. And per glucose molecule, we're going to see that you make 2 ATP during glycolysis. You make 0 ATP, so I'm going to write no ATP during pyruvate oxidation. And then in the Krebs cycle, you make another 2 ATP. And then in oxidative phosphorylation, you're going to make approximately 32 ATP. And oxidative phosphorylation is when chemoosmosis occurs. So the ATP that's produced during oxidative phosphorylation is going to be produced through chemoosmosis. And then the ATP that you produce during the Krebs cycle and glycolysis is produced through substrate level phosphorylation. Okay. Okay, so I previously told you that what we're going to do when we talk about the pathway is we're going to focus on the breakdown of glucose to provide the energy for the production of ATP. But it's important to know for this class that it's not the only energy source that can be broken down for this pathway. A cell can also break down proteins, carbohydrates, and fats, and those can all feed in to be fuel for aerobic respiration. They just happen to feed in at different points along the pathway. Okay, so you can break down proteins, carbohydrates, and fats in your food, and all of them can be used as the source of energy to produce ATP and aerobic respiration. Okay, but again, we're going to focus on this middle pathway that starts with glucose being broken down during glycolysis. Okay. So I just mentioned that we're going to focus on glucose as the energy source. And when we do that, this is going to be the equation that summarizes aerobic respiration. So this first molecule is glucose, C6H12O6 is glucose. You're also going to use oxygen, and then you're going to get the byproduct of carbon dioxide, a byproduct of water, and then you're going to get the 36 ATP, which is the goal of the whole pathway. So again, similar to the photosynthesis equation, you should be able to recognize this equation in a multiple choice type of question on an exam or quiz. Okay, so this I've already gone through. So when we talk about aerobic respiration, we're going to divide it into these 4 steps again: glycolysis, pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation. And I've also already discussed where they occur. So just to repeat, glycolysis in the cytosol, the last 3 in the mitochondria, pyruvate oxidation in the citric acid cycle, specifically in the matrix, and oxidative phosphorylation occurs at the inner membrane of the mitochondria. Okay. So this is just showing you in terms of a picture that glycolysis is happening in the cytosol, and then this is pyruvate oxidation, the Krebs cycle, and oxidative phosphorylation, all occurring within the mitochondria. Okay, so we're going to start with the first stage, glycolysis. And so the glyco part refers to, always refers to carbohydrate, and lysis obviously means to break or split. So what you're doing in glycolysis is you're splitting sugar. You're splitting glucose. So what's going to happen in the cytosol cells is that glucose is going to be broken down eventually into 2 pyruvate molecules. And then during that conversion of glucose, or that breakdown of glucose to 2 pyruvate molecules, you're going to produce 2 important things. You're going to produce those 2 ATP. You're also going to produce 2 NADH. Okay? And NADH is similar to NADPH in the sense that it's a molecule that carries electrons. And we'll talk about why it's important in this process later on. Okay, so when we talk about glycolysis, it actually consists of 10 reactions. But similar to the Calvin cycle, we're going to split up glycolysis into 3 phases. That each phase consists of multiple reactions. And for this class, you just have to know what happens during each phase, not what happens in each specific part of the pathway. Okay. So this has the stages on there. So we're going to divide it into the first stage is called the energy investment phase. The second stage is called the cleavage phase. And then the third stage is called the energy payoff phase. And so the first thing I'm going to do is tell you why they're called. It's going to be obvious once we go through it why they're called that. So the energy investment phase is called that because you actually have to use ATP during this phase. Okay, so rather than producing it, which is the goal of the pathway, you have to use 2 ATP, and that's going to be an energy investment that you get payoff later for. Okay. Then in the cleavage phase, that's the phase where you actually break apart a 6-carbon molecule into 2 3-carbon. So cleavage refers to cleaving something. So you're cleaving a 6-carbon molecule into 2 3-carbon molecules. And then the energy payoff phase is the phase where you produce the ATP that you make in this pathway, and you also produce 2 NADHs. So the payoff is the ATP and the NADH that you produce during that phase. That's the energy payoff. Okay, so this is just showing you that this detail you don't need to know, but the energy investment phase consists of 3 reactions, the cleavage phase consists of 2 reactions, and then the energy payoff phase consists of the remaining steps out of 10. Okay, so for this class, you do need to know what happens during each phase. So the first one we're going to talk about is the energy investment phase, which you can just write on the slide there. That's why I left it blank. And what you're going to do in the energy investment phase is you're going to start with glucose. And this is a set of 3 reactions. And over three reactions, you're going to produce a molecule called fructose, 1,6-bisphosphate. And that's the product of this phase. And the bisphosphate refers to the fact that you have 2 phosphate groups now attached to the molecule. And to get those 2 phosphate groups, you're going to have to use ATP. So during this step, you use 2 ATP. So that's 2 ATP feeding into this process where you produce fructose 1,6-bisphosphate. So remember, this is the investment that you're making. You're investing 2 ATP into this process. Okay, so that's the energy investment phase. And then you have the cleavage phase. And fructose 1,6-bisphosphate is the starting material for this phase. And so fructose 1,6-bisphosphate, like glucose, is a 6-carbon sugar. So fructose is also a 6-carbon sugar. So what's going to happen is you're going to break fructose 1,6-bisphosphate down into 2 glyceraldehyde 3-phosphate molecules. Okay, so in this cleavage phase, again, you take a 6-carbon molecule and you cleave it into 2 3-carbon molecules. And the names, so you start with fructose 1,6-bisphosphate, which is the 6-carbon molecule, and over the course of 2 reactions, you're going to split it into 2 glyceraldehyde 3-phosphate molecules. Okay, and then the last phase is the energy payoff phase. And this phase is going to start with the 2 glyceraldehyde 3-phosphate molecules that you made in the cleavage phase. So I'm just going to abbreviate glyceraldehyde 3-phosphate G3P. And this is going to consist of 5 reactions, and each glyceraldehyde 3-phosphate is going to be converted into a pyruvate molecule. So one of the products of this part of the pathway, of this stage or phase, is 2 pyruvate molecules. And then during this conversion, you're going to produce 2 NADHs, and I'll explain what's happening in terms of those NADHs in just a second. And you're also going to produce 4 ATP. Okay, so the products of the energy payoff phase are 2 NADHs, 4 ATPs, and 2 pyruvates. So back when we were listing out how much ATP each stage made, we said that glycolysis only produces 2, and that's because you have to subtract the 2 that you invested from the 4 that you produce. Okay, so because you invested 2 at the beginning, you only get a net of 2 ATP produced during glycolysis when you break 1 glucose molecule down. Okay, also what's happening in terms of NADH in this series of reactions is you're actually taking NAD plus and you're reducing it to NADH. So remember that when NAD plus is reduced to NADH, it's picking up electrons. Okay, so in this case, electrons are going to be taken from an organic molecule and given to NAD plus to reduce it to NADH. And then what we're going to see is that the NADH that's produced in glycolysis is going to be used in oxidative phosphorylation. Okay? So that's the very last stage of aerobic respiration. So again, the ATP, that's the goal of the pathway is to make that, but then we're going to see that the NADH that you produce is used in oxidative phosphorylation to produce even more ATP. Okay, so those are the three phases of glycolysis, all of them occurring in the cytosol. Do you guys have any questions about them?
So the NAD plus the NADH, just electrons basically are produced?
No, electrons are given to NAD+. So you would say NAD plus is what's reduced. So NAD plus is reduced to NADH. So I just make that distinction because you're not synthesizing a whole NADH molecule. All you're doing is taking NAD plus, you're adding 2 electrons and a hydrogen ion to it in the reduction process. And then, so basically you can think of this molecule here as an electron carrier. So it picks up electrons in glycolysis, and then it's going to carry those electrons to the electron transport chain that's used in oxidative phosphorylation. Okay, so those electrons are going to be used later on in aerobic respiration. Okay, and then just to remind you that the ATP made during this process is going to be made through substrate level phosphorylation. Now what I wrote in here, and you don't need to know, is I just put this in here if you just wanted to see them, but this is each of the reactions that has to take place in glycolysis. So this shows you the energy investment phase where you start with glucose. This is the first reaction, the second reaction, and the third reaction of the energy investment phase. So you start with glucose and you end with fructose 1,6-bisphosphate. And in this process, you're going to have to use 1 and 2 ATP molecules. So you invest 2 ATP molecules. Then this shows you the 2 steps of the cleavage phase. So you start with fructose 1,6-bisphosphate. In the end, you're going to end with 2 glyceraldehyde 3-phosphates. And then the energy payoff phase consists of steps 6 through 10. And you're going to produce 1 NADH here. You're going to produce 2 NADHs because you have 2 glyceraldehyde 3-phosphates made per glucose molecule. And then you're also going to produce 4 ATP total during this phase. Okay, also glycolysis is regulated by feedback inhibition. So if you remember back to energy enzymes and metabolism, we said that feedback inhibition, and again, sorry, let me reiterate, you do not need to know the individual reactions, you need to know the 3 phases. Okay? So the 10 reactions that are listed on the last 3 slides, you can ignore that unless you want to know the details. But you just need to know the 3 phases for this class. So back to the fact that glycolysis is regulated by negative feedback inhibition. What happens in feedback inhibition, if you remember from energy enzymes metabolism, is that one of the products of the pathway feeds back and inhibits an enzyme that acts earlier in the pathway. And for this pathway, ATP is actually a non-competitive inhibitor of phosphofructokinase, which if you look at these reactions, phosphofructokinase catalyzes the 3rd step. So what happens is ATP is a non-competitive inhibitor of this enzyme. And so it'll stop the pathway in that way. Okay, do you guys have any questions about what we've discussed for glycolysis, which is the first, in terms of aerobic respiration, glycolysis again is the first stage of aerobic respiration. Okay, so the second part of, or second stage of aerobic respiration is going to be pyruvate oxidation. In the first bullet point, it says that basically you can think of pyruvate oxidation as connecting glycolysis to the citric acid cycle. So what we're going to see happens during pyruvate oxidation is you start with the products of glycolysis, the product of glycolysis, 2 pyruvate molecules. So you're going to start with the 2 pyruvate molecules that you produce in glycolysis. And then during pyruvate oxidation, each of those pyruvate molecules are going to end up becoming an acetyl coenzyme A molecule. And acetyl coenzyme A is going to be the starting material for the Krebs cycle. So that's how it connects glycolysis to the citric acid cycle. Okay, so remember we said that pyruvate oxidation occurs in the mitochondrial matrix. So for pyruvate oxidation, the pyruvate that you produce in glycolysis is made in the cytosol, and the first thing that's going to have to happen is it's going to have to be transported into the mitochondrial matrix. Once it's in the matrix, then pyruvate oxidation can occur. And this stage is catalyzed by a multi-enzyme complex called pyruvate dehydrogenase complex. So pyruvate dehydrogenase complex is a multi-enzyme complex that catalyzes this stage. And it actually consists, this detail you don't need to know, the multi-enzyme complex consists of 3 different types of enzymes that catalyze 3 separate reactions, but all of them are part of the pyruvate oxidation process. Okay? And so essentially what's going to happen during this process is you're going to take the 2 pyruvates that you made in glycolysis, and each of them is going to be broken down, and they give off carbon dioxide. So one thing you're going to produce is 2 carbon dioxides. You're also going to produce 2 NADH molecules. And again, during this phase, what you're doing is you're taking NAD plus and you're reducing it to NADH. So it's picking up, NAD plus is picking up electrons in this stage. Okay, and then what you're going to be left with is at the end of this stage is 2 acetyl coenzyme A's, and we just abbreviate coenzyme A, CoA. Okay, so the products of this stage are NADH, carbon dioxide, and 2 acetyl coenzyme A's. What we're going to see is that acetyl coenzyme A is the starting material for the next stage, which is the Krebs cycle. The carbon dioxide that you produce is a waste product, and in us that's what you would exhale that. And then the NADH that you produce during this stage is also going to be used in the last stage of oxidative phosphorylation, just like the NADH that you produce during glycolysis. Okay, so the NADH that you produce is going to be used in oxidative phosphorylation to produce ATP. Okay, so you don't need to know these specific details that I've written on this slide here, but it actually just breaks down the 3 reactions that pyruvate dehydrogenase complex catalyzes. So first of all is it removes a carboxyl group, which gets released as carbon dioxide. Then the 2 carbon molecule that you are left with is oxidized, which means you take electrons from it. The electrons that are taken from that 2 carbon molecule are used to reduce NAD plus to NADH. And then what you do is you take that oxidized molecule and you attach it to acetate, and that gives you acetyl coenzyme A. So those are the 3 reactions that are catalyzed by the pyruvate dehydrogenase complex. I just want you to know again the overall process over here. What do you start with, what do you end with, and what do you produce during the process. Okay, so per glucose molecule, pyruvate oxidation is going to produce 2 NADHs, 2 carbon dioxides, and 2 acetyl coenzyme A's. Okay, so we said that the acetyl coenzyme A's are going to be the starting material for the Krebs cycle or the citric acid cycle. And the citric acid cycle is called a cycle for the same reason that the Calvin cycle is called a cycle. So in the citric acid cycle, you're going to take the acetyl group. So the first step of the Calvin cycle, you're going to take the acetyl group from acetyl coenzyme A and attach it to oxaloacetate. That's the first step. So you take the acetyl group, attach it to oxaloacetate. That's the first step. And then at the end of 7 more steps, you get oxaloacetate back. So in this cycle, you regenerate oxaloacetate, and that's why it's called a cycle for this particular pathway. So you need to know the first step, which I just described to you. You take the acetyl group and you attach it to oxaloacetate, and that produces a molecule called citrate. So you need to know that first step. You take the acetyl group from acetyl coenzyme A, attach it to oxaloacetate, that produces citrate. And then what happens is over the course of 7 reactions, you take citrate and regenerate oxaloacetate. Okay? Now, keep in mind that per glucose molecule, you're going to get 2 acetyl coenzyme A's entering this cycle. So what that means is we can describe it as the cycle turning twice per glucose molecule. So basically everything you see happening in that pathway happens twice per glucose molecule. So that means that you're producing, so you can count up here that you have 1, 2, 3 NADHs being produced per turn of the cycle. So that means in total you're going to get 6 NADHs produced. And again, that's NAD plus being reduced to NADH. And that's going to happen 6 times during the cycle, per glucose molecule. You can see that you have 1 FADH2 molecule being produced. So per glucose molecule, that means this cycle is going to produce 2 FADH2 molecules. And FADH2 is similar to NADH in the sense that it carries electrons. So what's happening during this cycle is you're taking FAD and you're reducing it to FADH2. You can also see that in 1 turn of the cycle, you're going to produce ATP. So for per glucose molecule, this cycle produces 2 ATP. You can also see that for 1 turn of the cycle, you're going to produce 2 carbon dioxides, which means that per glucose molecule, you're going to produce 4 carbon dioxides during the Krebs cycle. So these are the important products of the Krebs cycle per glucose molecule. 6 NADHs, 2 FADH2s, 2 ATPs, and 4 carbon dioxides. Okay, so starting from the bottom and going up, again, carbon dioxide is a waste product of this process. ATP, molecule you're trying to make. And then the FADH2 and the NADH that you produce in this cycle are also used in oxidative phosphorylation to produce more ATP. Okay, so the NADH and FADH2 molecules that you're making in each of the stages, you're going to be using later in oxidative phosphorylation. Okay, and then again, this cycle is happening in the mitochondrial matrix within the mitochondria. Okay, do you guys have any questions about this particular stage of aerobic respiration?
The cycle because
Oxaloacetate?
yeah, it's produced again, basically like it's
Yeah. Yeah.
it's like-
Yeah. Okay. This just shows you the individual steps of the Calvin cycle. You don't need to know the individual steps. But just to point out, to relate this to something that we've learned about, we learned about competitive versus non-competitive inhibitors and feedback inhibition. And the Krebs cycle is also regulated by feedback inhibition. And it turns out that, oxaloacetate is a competitive inhibitor of succinate dehydrogenase. So that means that oxaloacetate binds to the active site of that enzyme and inhibits it. And that would be a form of feedback inhibition. Okay, so the last stage and probably the most complicated to understand is oxidative phosphorylation. The different things that you're going to need for oxidative phosphorylation is you're going to need the NADH and FADH2 molecules that you made in the previous stages, and they're going to be providing the electrons that you need for this process. So the electrons that they picked up in the previous stages are going to be used in this stage. You're also going to need an electron transport chain, which is going to be located in the inner mitochondrial membrane. And we'll state this now, but the importance of the electron transport chain is it creates a hydrogen ion electrochemical gradient. So we're going to see that the electron transport chain produces a hydrogen ion electrochemical gradient. And it's important for the same reason that it was important in photosynthesis, that the hydrogen ion electrochemical gradient that's produced by the electron transport chain is the power source for ATP synthase. And ATP synthase is obviously going to be the enzyme that makes ATP during oxidative phosphorylation. And so this is chemiosmosis because you're using an electrochemical gradient as the power source to make ATP during this process. Okay, and then what we're also going to see is that the final electron acceptor, so we said electron transport chains pass electrons from component to component. The final electron acceptor at the end of this chain is oxygen. And that's where you actually consume oxygen in aerobic respiration. So we said that oxygen is required for aerobic respiration. It's specifically required for this last stage where it's the final electron acceptor. That's where it gets consumed. So the oxygen that you breathe is important because it's the final electron acceptor in this process. Okay, so I just want to reiterate something that I've already stated to you, and that's that during oxidative phosphorylation, ATP synthase is the enzyme that makes ATP, and the power source for ATP synthase is going to be a hydrogen ion electrochemical gradient, and again, that gradient is created by the electron transport chain. So if you were to do something to inhibit the electron transport chain, which is what cyanide does, you would inhibit the production of ATP by ATP synthase during this process. Because if you inhibit the electron transport chain, that means you do not get the hydrogen ion concentration gradient, and that means then there's no power source for this enzyme and it can do nothing. Okay, so if you do something to prevent the hydrogen ion electrochemical gradient from being created, that's going to prevent this enzyme from working. Okay, so, and then you have a hydrogen ion electrochemical gradient, which, if you remember from energy enzymes, or sorry, not that, membrane function, structure, and transport, an electrochemical gradient is the combination of a membrane potential and a concentration gradient. And this electrochemical gradient exists across the inner membrane. So what they're showing you here is this is the inner membrane of the mitochondria, and this picture is trying to describe both the membrane potential and the concentration gradient and the direction of movement in terms of what it favors. Okay, so basically the important thing for this class is that in terms of the hydrogen ion electrochemical gradient that gets created during this process, you create a concentration gradient where you have a higher concentration of hydrogen ions in the intermembrane space relative to the matrix. Okay, so what that means is the hydrogen ions, in order to power ATP synthase, need to flow through it like they did in photosynthesis. And so in this process, ATP synthase is sitting in this membrane, and hydrogen ions are going to flow through it from the intermembrane space into the matrix. Okay, and what we're going to see in a second is that the electron transport chain pumps hydrogen ions in the opposite direction. So the electron transport chain creates this concentration gradient by pumping hydrogen ions in the intermembrane space, and then they can flow back through ATP synthase to power that synthase to make ATP. Okay. Previously I have described the electron transport chain to you. So you have a series of components that make up the electron transport chain, and electrons are going to be passed from component to component in a very specific order in terms of the components pass electrons specifically to, so component 1 specifically passes to 2, 2 passes to 3, and so on. And what happens is as electrons are being passed from component to component, they're losing energy. So the electrons are going to be giving up energy during this, we're going to call it electron flow. And the energy that's released during this movement can be used to power different things. And during this process, it's specifically used to power the pumping of hydrogen ions. Okay, so pumping hydrogen ions, moving something against this concentration gradient, is going to require energy. And in this case, the pumping of hydrogen ions is going to be powered by the electrons. So as the electrons flow through this electron transport chain, energy is released, and the energy released powers the pumping of hydrogen ions into the intermembrane space. Okay, there's not really anything important, so we'll go through these in a second, but this is showing you the different components of the electron transport chain. So, and this is specifically showing you NADH, but FADH2 would also be passing its electrons to the electron transport chain, but it's showing you that the electrons, as they flow from NADH to oxygen, which is the final electron acceptor, they're going to be losing energy. So they're releasing energy. So this is an exergonic process. So this electron flow is exergonic. And again, the energy that's released during this process is used to pump hydrogen ions specifically into the intermembrane space. That creates the hydrogen ion electrochemical gradient that powers the ATP synthase that's also embedded in the inner membrane, and the hydrogen ions are going to flow through that synthase, powering it to synthesize ATP. Okay. So what we're going to see happens in this process is that it's going to start with NADH and FADH2 donating their electrons to the electron transport chain. Then the electrons flow along the electron transport chain. That pumps hydrogen ions in the intermembrane space, creating a hydrogen ion concentration gradient. The electrons that are flowing down the electron transport chain are going to end up being added to oxygen. And then the hydrogen ion concentration gradient that you create is going to be used to power ATP synthase. Okay? So you do need to know the different components of this electron transport chain. One of the components is called NADH dehydrogenase complex. And all of these components are either embedded in the inner mitochondrial membrane or they're associated with the inner mitochondrial membrane. So this is all happening at the inner mitochondrial membrane. The second component that you need to know is succinate reductase. The third component of this electron transport chain is the cytochrome BC1 complex. A fourth component is called cytochrome oxidase. And then two more components are ubiquinone and then this isn't written in the order of necessarily of electron flow. I'm just listing out the components. The last component is cytochrome C. Okay. On the slide here, I mentioned that some of the components are multi-protein complexes, so 4 of them. The multi-protein, I'm not going to ask you this on a quiz or exam, but the multi-protein complexes are 1 through 4 here. So NADH dehydrogenase succinate, cytochrome BC1, cytochrome oxidase are your multi-protein complexes. And then the electron transport chain also includes these 2 components, one called ubiquinone and the other called cytochrome C. In terms of these components, so they're all components of the electron transport chain. I want to tell you one more thing, and then we'll take another 10-minute break. the NADH dehydrogenase complex, the cytochrome BC1 complex, and cytochrome oxidase are not only components of the electron transport chain, they're also hydrogen ion pumps. So when electrons pass through these complexes, these are the complexes that actually do the job of pumping hydrogen ions into the intermembrane space. Okay? So NADH dehydrogenase complex, cytochrome BC1 complex, cytochrome oxidase, as electrons pass through these complexes, they're going to pump hydrogen ions into the intermembrane space. Okay, so let's take a 10-minute break now, and what we'll do is we'll talk about, I'll use the board again to talk about the pathway of electron flow, and then I'll use the slides to describe the same process. So